So this is some solution that we're going to put that's going to be going into the inside of the cell.
At UT Southwestern's Department of Pharmacology, Dr. Rama Ranganathan is shedding light on how proteins carry out vitally important chemical reactions in cells. Proteins are made up of amino acids, which seem to interact with each other like a tightly packed 3D jigsaw puzzle. Rama developed a new computational model revealing which amino acids are most critical for the structure and function of proteins.
We are looking at all the products, all the proteins that have survived over the ages through the process of evolution. And by looking at that ensemble of sequences from all the species, we can discern the basic rules by which the protein is made.
Rama's computational method showed the existence of tiny networks of amino acids that link distant sites on proteins. These networks allow proteins to communicate, carrying signals between different points like the wires in electrical circuits. Defects or mutations in these amino acid networks can trigger cells to behave improperly, causing diseases such as cancer or diabetes. Rama's computational method could potentially be applied to find out which sites in proteins are most sensitive to these mutations.
Understanding that allows us to begin to perhaps rationally design medicines or drugs that will alter that mutation so that it can now function perhaps.
Rama's model was new and different and initially had its skeptics.
Over 100 laboratories have taken this computational method that he's discovered and started to test them in their laboratories. And in every case that's been tested so far, it's stood the test.
Rama and his team more recently applied his computational method to an even more rigorous test, building a synthetic protein from scratch. Their result was several artificial proteins that showed the same structure and function as natural ones.
Being able to design a protein from scratch is I think, a significant step because it means that the rules that we learned, we're on the right track.
Ultimately, these models for proteins have to be tested in living organisms to show that they can work in a natural setting. To do this, Rama and his team are working with bacteria, yeast, and flies to understand the ways natural and designed proteins may function differently in cells.
This is a fly that carries a mutant form of a protein.
Rama and other scientists are also testing his models for protein function using the neuronal cells in the eye that use a network of proteins to sense light. It's the first step in vision.
The neuron would then send the output out of this end of the cell to the rest of the brain.
Someday, the research taking place in Rama's lab could lead to new personalized medical strategies.
It's becoming evident that his work has the possibility to completely revolutionize how we understand protein architecture.
While Rama knows these goals are years down the road, he enjoys every discovery he makes along the way.
We have all these fantasies about big questions we want to address, but actually just the ability to contribute to these questions in some non-trivial way is already a success in my view.